This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to Genetic Leaps: Rewriting Evolution's Rules. For generations, we've pictured life's evolution as a grand tree, with genes flowing faithfully downwards. But what if there's a secret, dynamic highway where life shares its fundamental information sideways? This narration will explore horizontal gene transfer, or HGT, a phenomenon radically reshaping our understanding of evolution. Get ready to discover a biological landscape far more interconnected and fluid than you ever imagined.
Chapter 1: Genetic Leaps: Rewriting Evolution's Rules. For generations, our understanding of life's evolution has been rooted in the concept of a grand tree, with branches steadily diverging from a common ancestor, each species faithfully passing its genetic blueprint down through generations. This process, known as vertical gene transfer, is how offspring inherit DNA from their parents, ensuring the continuity and gradual adaptation of species over vast stretches of time. But what if this isn't the whole story? What if there's another, more dynamic and utterly transformative way that life shares its most fundamental information? Imagine a world where genetic material doesn't just flow downwards, but leaps sideways, across species, across kingdoms, even across the deepest chasms of the tree of life. This phenomenon is precisely what scientists call horizontal gene transfer, or HGT, and it's radically reshaping our understanding of evolution, revealing a biological landscape far more interconnected and fluid than we ever imagined. It's the movement of genetic material between organisms not by reproduction, but by direct or indirect means, making it a powerful, rapid engine for change and adaptation across the living world.
The first whispers of this extraordinary process emerged long before we fully understood DNA itself. Back in 1928, a British bacteriologist, Frederick Griffith, observed something remarkable. Harmless bacteria could become deadly simply by being exposed to the remains of heat-killed pathogenic bacteria. He called it a transforming principle, a mysterious agent that could pass genetic information between bacterial strains. Years later, in the late 1930s and early 1940s, Oswald Avery, Colin MacLeod, and Macklin McCarty definitively identified this principle as DNA, and the process was transformation, one of the key mechanisms of horizontal gene transfer. Further groundbreaking observations followed. In 1951, researchers discovered that a viral gene, when transferred into a harmless bacterium called Corynebacterium diphtheria, could turn it virulent, revealing not only the mechanism behind diphtheria's sudden onset of symptoms, but also how patients could carry the infection asymptomatically before a sudden conversion to virulence. This discovery marked the first example of the lysogenic cycle's relevance to bacterial pathogenicity. Then, in 1959, a pivotal discovery in Japan demonstrated the transfer of antibiotic resistance between different species of bacteria. These early insights laid the groundwork for what would eventually be recognized as a pervasive evolutionary force, one that challenges the traditional, strictly vertical view of life's history. By the mid-1980s, some scientists were already postulating that this lateral gene transfer had been a significant factor since the very dawn of life on Earth, shaping all of evolutionary history.
So how exactly does this genetic leap happen? Scientists have uncovered several ingenious mechanisms, particularly well understood in the microscopic world of bacteria and archaea. One primary method is transformation, where a bacterium directly takes up naked DNA from its environment. This DNA, often released by other cells that have died and lysed, simply floats freely in the surrounding medium, and indeed, naked DNA and RNA are known to circulate in various bodily fluids and environments. But not all bacteria can do this at will.
For a bacterium to bind, take up, and incorporate this foreign DNA into its own chromosome, it must enter a special physiological state known as competence. For example, in a common bacterium like Bacillus subtilis, developing this competence requires the expression of about 40 specific genes. This remarkable ability, essentially allowing a cell to scavenge and integrate new genetic information, is a common occurrence in laboratories, where it is harnessed to insert novel genes into bacteria for various experiments or for industrial and medical applications. Another fascinating mechanism is transduction, a process mediated by viruses that specifically infect bacteria, known as bacteriophages, or simply phages. In transduction, a phage acts as an unwitting messenger, carrying bacterial DNA from one bacterium to another. During its replication cycle within a host bacterium, the phage can sometimes accidentally package fragments of the host's bacterial DNA into its viral particles, instead of, or in addition to, its own genetic material. When this hybrid phage then infects a new bacterium, it injects this bacterial DNA, which can then be integrated into the recipient's genome. This can occur in a generalized way, where any piece of bacterial DNA might be transferred, or in a more specialized manner, typically involving genes located near where a phage's own genome has integrated into the bacterial chromosome. The sheer abundance of phages in many environments, from the human gut to the vast oceans, means this mechanism is a powerful and constant driver of genetic exchange. Perhaps the most well-known mechanism, especially for the rapid spread of crucial traits like antibiotic resistance, is conjugation. This involves the direct transfer of DNA from one bacterial cell, the donor, to another, the recipient, through physical cell-to-cell contact. This contact is often established by a specialized, hollow tube-like structure called a conjugation pilus. The genetic material transferred during conjugation is typically in the form of a plasmid, a small circular piece of DNA that exists independently of the main bacterial chromosome and can carry a variety of genes, including those for antibiotic resistance. During conjugation, a type 4 secretion system, or T4SS, is used to passage DNA from the donor to the recipient. Interestingly, proteins that help stabilize single-stranded DNA, often encoded within these conjugative plasmids, are thought to play a role in this transfer, though some studies show they aren't always essential. This process is not limited to plasmid transfer. In certain bacteria, like Mycobacterium smegmatis, conjugation can lead to extensive blending of parental chromosomal genomes, a process that strikingly resembles aspects of sexual reproduction. Beyond these classical three, other mechanisms further expand the web of genetic exchange. Gene transfer agents, or GTAs, are peculiar virus-like elements produced by some bacteria, such as those in the Rhodobacterales order. These tiny particles are encoded by the host cell itself and act as natural vectors, transferring random fragments of the host's own DNA to other cells. This suggests a host-controlled mechanism for distributing genetic information within a community.
Then there's horizontal transposon transfer, or HTT. Transposable elements, often called jumping genes, are mobile segments of DNA that can move from one location to another within a genome. Crucially, they can sometimes pick up other genes, like those conferring resistance, and insert them into a plasmid or the main chromosome, thereby facilitating HGT. Evidence suggests that HTT is not just crucial for prokaryotic evolution, but is also a common and widespread phenomenon in eukaryotes. Certain types of transposable elements, like DNA transposons and LTR retroelements, are more likely to undergo HTT due to their stable double-stranded DNA intermediate. While the exact vectors for these genetic leaps are still being investigated, proposed mechanisms include arthropods, viruses, freshwater snails, and various endosymbiotic or intracellular parasitic bacteria. This mobility can introduce new genetic material, shuffle genes, and even lead to new functions being co-opted by the host genome. The impact of horizontal gene transfer is profound and ubiquitous, observed across all domains of life. In prokaryotes, bacteria, and archaea, HGT is exceptionally common, even between very distantly related organisms. It's a driver for the spread of critical traits like antibiotic resistance and virulence factors. HGT also empowers bacteria to break down novel compounds, including human-made pesticides, and plays a significant role in the production of specialized metabolites, as seen in groups like P. aeruginosa and Actinomycetalis, whose polyketide synthases are modular and easily exchanged. Even archaea, like the haloarchaea, have harnessed HGT, acquiring numerous bacterial genes to adapt to extremely salty environments. Other archaea, such as Sulfolobus sulfatericus and Sulfolobus acidocultarius, demonstrate a unique response to DNA damage from UV radiation, inducing cellular aggregation and facilitating intercellular DNA exchange, which appears to be a crucial mechanism for DNA repair through homologous recombination, a primitive form of sexual interaction. HGT also occurs in eukaryotes, demonstrating the fluidity of genetic information across the tree of life. For instance, mechanisms like grafting in plants can facilitate the transfer of chloroplasts, mitochondrial DNA, and even entire cell nuclei between species. These examples merely scratch the surface of the incredible genetic exchanges occurring across kingdoms. Chapter 3 will delve into these interkingdom transfers in much greater detail, revealing the full extent of these unseen genetic highways. This ever-growing body of evidence reveals a biological landscape far more dynamic and interconnected than previously imagined, where genetic information flows across boundaries once thought impenetrable. Horizontal gene transfer challenges the traditional, strictly vertical model of evolution, revealing a more fluid and adaptable biological reality. It represents a powerful, rapid engine for change, introducing novel traits, boosting adaptability, and allowing organisms to colonize new niches. HGT significantly contributes to the genetic variation that fuels natural selection, often providing a quicker route to adaptation than relying solely on mutations. It compels us to fundamentally rethink the rules of evolution and the very definition of species, especially in the microbial world where genetic exchange is rampant. The implications of this pervasive genetic exchange are far-reaching, presenting both an incredible engine for biological innovation and, as we will explore, significant challenges for life on Earth. How will this deeper understanding of HGT continue to reshape our view of life's grand narrative? Chapter 2: HGT, Fueling the Resistance Crisis. Today, we delve into one of the most urgent and alarming consequences of horizontal gene transfer, HGT, its role in the escalating global crisis of antibiotic resistance. Antibiotic resistance isn't just a medical inconvenience, it's a profound threat to global health, undermining our ability to treat common infectious diseases. What makes this crisis so intractable is the incredible speed and efficiency with which bacteria can acquire and spread resistance genes. While mutations certainly contribute to a bacterium's ability to withstand antibiotics, this process is generally slow and often results in harmful changes. HGT, however, allows bacteria to acquire entirely new genes, or even large segments of DNA, from other organisms in a single, rapid exchange. This allows bacteria to acquire entirely new resistance genes at an astonishing pace, far outstripping our ability to develop new antibiotics. Scientific consensus now identifies HGT as the primary mechanism driving the widespread dissemination of antibiotic resistance among bacteria, profoundly influencing the evolution of pathogens and posing immense challenges for modern medicine. This is not merely about new resistance emerging. Many resistance genes evolved long ago in natural environments with no anthropogenic influence. But HGT is now rapidly spreading these pre-existing genes to and among human pathogens. Let's delve into how the mechanisms of HGT are specifically harnessed to fuel this resistance crisis. Perhaps the most significant player in the rapid spread of antibiotic resistance is conjugation.
This process is a highly efficient way to transfer large pieces of genetic material, particularly plasmids. These independent DNA molecules are notorious for carrying genes that confer antibiotic resistance. A single bacterium that develops resistance can, through conjugation, rapidly arm an entire population, even different species of bacteria, against an antibiotic. Factors such as the plasmid's repression status, cellular density, and temperature are crucial in influencing the frequency of this transfer. Consider the ongoing fight against Staphylococcus aureus, a bacterium responsible for a wide range of infections. The emergence of methicillin-resistant S. aureus, or MRSA, is a stark example of HGT in action. Studies have shown that a particular MRSA strain, CC398, acquired its methicillin resistance after being exposed to common antibiotics like tetracycline and beta-lactams, often used in agriculture. Once resistance genes are acquired through HGT, these bacteria don't just stop there. They proliferate and continue to evolve, moving swiftly among patients and throughout hospital environments, leading to the development of diverse and highly persistent populations, such as the widely recognized USA 300 strain. The rapid evolution of Mycobacterium tuberculosis, a formidable pathogen, also showcases conjugation's impact, as it has been linked to the acquisition of antibiotic resistance genes through this very mechanism. The sheer prevalence of conjugative plasmids as vectors for HGT in bacteria makes them a prolific force in the spread of resistance, directly impacting clinical outcomes and public health. This continuous genetic arms race means that even new drugs and diagnostic tools are constantly challenged by the persistence of HGT, forcing practitioners and researchers into a race against bacterial evolution. Transduction, mediated by bacteriophages, also plays a critical role in disseminating resistance. During their life cycle, these phages can sometimes mistakenly package fragments of bacterial DNA, including antibiotic resistance genes, into their viral capsids. When these phages infect new bacteria, they deliver this genetic payload, effectively transferring resistance. This mechanism is so pervasive that phages are increasingly recognized as significant reservoirs of antibiotic resistance genes in various environments. Beyond resistance, transduction also spreads virulence factors, exacerbating the global health crisis. Recent research further illuminates the complex interplay between environmental stressors and phage-mediated HGT. Heavy metal contamination, which is widespread in agricultural soils, has been identified as a potent co-driver of resistance. Studies reveal that lysogenic phages, those that integrate their genetic material into the host bacterium, act as crucial mediators in this process. Under heavy metal stress, these phages can promote the dissemination of antibiotic resistance genes. They do this through two potential mechanisms. First, by encoding auxiliary metabolic genes that reprogram host metabolism, enhancing bacterial survival and adaptation, thereby facilitating the co-transfer of adjacent resistance genes. Second, phage-encoded heavy metal detoxification genes directly mediate metal detoxification, driving the co-transfer of neighboring resistance fragments, and inducing changes in membrane permeability, which collectively enhance gene mobilization. This means that environmental pollutants can indirectly accelerate the spread of antibiotic resistance by leveraging phage activity, revealing an often overlooked pathway linking environmental contamination to resistance propagation. Then there's transformation, where bacteria take up DNA directly from their surroundings. While often considered less common in nature than conjugation, it's a vital pathway for genetic exchange, especially for species that have evolved a state of competence. In the context of antibiotic resistance, bacteria can scavenge resistance genes released by dead or degraded cells, integrating them into their own genomes. This is particularly relevant in environments where bacterial cell density is high, or under conditions of nutritional limitation, which can induce competence in many bacterial species. Beyond these three classical mechanisms, other elements significantly amplify the problem.
Transposable elements are mobile DNA segments. These elements can pick up resistance genes and insert them into plasmids or directly into the bacterial chromosome, thereby directly inducing HGT of antibiotic resistance. For example, specific insertion sequences, such as IS3 and IS30, have been found adjacent to the BLA and DM1 gene cluster, a notorious resistance gene conferring resistance to nearly all clinical beta-lactam antibiotics. Their presence indicates a clear risk of transposon-mediated HGT, contributing to the global spread of BLA and DM1 carrying bacteria like Klebsiella, Escherichia, Acinetobacter, and Pseudomonas species, particularly prevalent in hospital environments and wastewater. Another crucial mobile genetic element is the integron. Integrons are genetic platforms capable of capturing and expressing genes, particularly antibiotic resistance genes, by site-specific recombination. They are considered the most important way for the dissemination and diffusion of antibiotic resistance genes between bacterial strains, acting as efficient gene capturing systems that can rapidly assemble multiple resistance genes into a single mobile unit.
The environment plays a critical, often underestimated role in accelerating the spread of resistance genes through HGT. Antibiotic use, both in human medicine and agriculture, creates a powerful selective pressure, favoring resistant bacteria and driving the transfer of resistance genes. But it's not just antibiotics. Disinfectants, widely used in industrial and healthcare settings, also contribute significantly. When bacteria encounter disinfectants, they can increase their production of efflux pumps, molecular machines that actively pump out harmful substances, including many antibiotics. This leads to a dangerous co-resistance, where bacteria become resistant to both disinfectants and antibiotics simultaneously. Crucially, scientific evidence demonstrates that chlorination, a common disinfection method, increases horizontal gene transfer rates. This occurs through the combined effects of increased membrane permeability and oxidative stress, thereby facilitating the exchange of antibiotic resistance genes among bacterial species. This creates a vicious cycle where efforts to sterilize environments inadvertently foster greater resistance. The consequences of HGT-driven antibiotic resistance are profound and far-reaching. It leads to the rapid emergence of multi-drug-resistant strains, making once-treatable infections incredibly difficult, if not impossible, to manage. For example, the BLAND5 gene, which confers carbapenem resistance, can be transmitted between bacteria and packaged into active enzymes by outer membrane vesicles, or OMVs. These nanoscale proteoliposomes, secreted by gram-negative bacteria, can encapsulate both the resistance-bearing plasmids and the catalytically active enzymes, enabling the horizontal transfer of functional resistance to susceptible recipients. This acquisition can dramatically increase a recipient bacterium's resistance to powerful antibiotics like meropenem by over 500-fold, while the OMV-associated enzymes can even degrade antibiotics in the extracellular environment, protecting nearby susceptible bacteria. This highlights a sophisticated and underappreciated mechanism of resistance dissemination, creating a complex challenge for treatment strategies. Furthermore, HGT can transfer clusters of virulence genes known as pathogenicity islands, which can transform benign bacteria into dangerous pathogens. These islands, often containing multiple virulence factors, can be transmitted to other bacteria, reinforcing the multifaceted threat of the antibiotic resistance crisis by simultaneously spreading resistance and enhancing bacterial harmfulness. The rapid and unpredictable evolution of bacterial populations driven by HGT poses a significant problem for clinical surveillance and treatment. It necessitates individualized screening to determine effective therapies and detect new strains, such as high-level vancomycin-resistant S. aureus, or VRSA. This constant evolution means that even as new drugs emerge, the threat of resistance remains, requiring continuous vigilance and innovative approaches to stay ahead. Moreover, the human microbiome harbors a plethora of antimicrobial genes. If these genes are transferred to pathogenic microbes, the consequences for human health could be detrimental, turning commensal bacteria into dangerous adversaries. The phenomenon of horizontal gene transfer transforms our understanding of bacterial evolution, making it clear that pathogens are not isolated entities evolving in a vacuum. Instead, they are part of a vast, interconnected genetic network, constantly exchanging information, adapting, and innovating. This dynamic genetic landscape, particularly as it pertains to antibiotic resistance, presents an urgent call to action, compelling us to consider novel strategies that go beyond simply developing new drugs. While bacteria are adept at acquiring new genes, they also possess sophisticated defense mechanisms, such as CRISPR-Cas adaptive immune systems, to degrade foreign DNA, including conjugative plasmids. Understanding this evolutionary arms race, including both the mechanisms of HGT and the bacterial defenses against it, could unlock novel strategies for intervention. What will it take to truly understand and combat this unseen, yet alarming force shaping our fight against infectious diseases?
Chapter 3: Unseen Genetic Highways, Reshaping Eukaryotic Life. We've journeyed through the foundational mechanisms of horizontal gene transfer and witnessed its alarming impact on the spread of antibiotic resistance in the microbial world. But the story of HGT doesn't end with bacteria and archaea. Prepare to explore an even more astounding frontier, the unseen genetic highways that allow genes to leap across the vast evolutionary chasms separating entirely different life forms, profoundly reshaping the development and adaptation of eukaryotes, from fungi and plants to insects and even ourselves. For a long time, the prevailing scientific view held that complex multicellular eukaryotes, with their neatly compartmentalized cells and distinct reproductive processes, were largely immune to the kind of rampant genetic exchange seen in prokaryotes. Our understanding of evolution in these organisms focused almost exclusively on vertical gene transfer, the faithful inheritance of DNA from parent to offspring. Yet, as our genomic technologies have advanced, unraveling the intricate sequences of countless eukaryotic genomes, a revolutionary picture has begun to emerge. Advances in HGT detection, aided by next-generation sequencing, have demonstrated that HGT occurrence may have been previously underestimated. Growing evidence indicates that horizontal gene transfer, far from being a rare anomaly, is relatively common among many eukaryotic species, playing a significant, if often hidden, role in their adaptation to novel environments. The complexity of eukaryotic genomes, with their larger size and abundance of repetitive regions, has historically complicated the accurate identification and characterization of these transferred genes, making them truly unseen genetic highways until recently.
One of the most ancient and profound examples of inter-kingdom gene transfer lies at the very heart of eukaryotic cells, the origin of mitochondria and chloroplasts. According to the endosymbiotic theory, these vital organelles, responsible for energy production and photosynthesis, once existed as free-living bacteria. Billions of years ago, they were engulfed by a progenitor eukaryotic cell, forming an enduring partnership. Over eons, a massive horizontal gene transfer event occurred, with countless genes migrating from the genomes of these bacterial endosymbionts and integrating into the host cell's nuclear genome. This ancient genetic highway paved the way for the evolution of all complex life as we know it. But the story doesn't stop there. Even today, genes continue to move between organelles. For instance, the mitochondrial ATP1 gene has been reported to transfer between the parasitic genera Cuscuta and Bartzia and several Plantago species. Similarly, mitochondrial genes can transfer from host plants to parasitic plants like those in the Rafflesiaceae family, or chloroplast genes can move to the mitochondria of a bean plant. These are not just historical events. They are ongoing examples of genetic material flowing across cellular boundaries within a single organism, reflecting a deep evolutionary legacy of genetic exchange. Mechanistically, studies have even found a transmembrane potential-dependent mechanism of DNA uptake into plant mitochondria, likely involving a voltage-dependent anion channel. Beyond these ancient internal transfers, mobile genetic elements play a crucial role in horizontal transposon transfer, or HTT, facilitating genetic leaps between distinct species and even kingdoms. Their specific role in eukaryotic evolution is increasingly recognized. For instance, in plants, certain LTR retrotransposons of the Copia superfamilies, especially those with low copy numbers from the Ale and Ivana lineages, have been shown to transfer horizontally between different plant species. Both spatial and taxonomic proximity of species has been proposed to favor HTTs in plants and animals. In plants, the interaction between lianas and trees has been shown to facilitate HTT in natural ecosystems. In the animal kingdom, HTT has been observed across continents, with proposed vectors including arthropods, viruses, freshwater snails, and various endosymbiotic or intracellular parasitic bacteria. The arrival of a new transposon in a host genome can be disruptive, inducing mutations, but it can also be incredibly beneficial, introducing new genetic material, shuffling genes, and even leading to the host genome co-opting these elements to perform entirely new functions. The detection of HTT remains challenging due to its dynamic nature and the limited number of species analyzed, suggesting its true prevalence may be underestimated. Viruses, too, serve as powerful intermediaries in these cross-kingdom genetic exchanges, extending beyond their well-known role in bacterial transduction. Consider the intriguing case of Sputnik, a tiny virus that infects amoebae. Sputnik cannot reproduce on its own. It requires another virus, mimivirus, to have already infected the same amoeba cell. What's fascinating is that Sputnik's genome contains genes closely related to those of mimivirus and mammavirus, suggesting it might have cannibalized these genes during its history. This example reveals a horizontal gene transfer pathway between viruses, paralleling how bacteriophages ferry genes between bacteria. Even more broadly, bacteriophage-mediated mechanisms have been proposed to transfer genes between prokaryotes and eukaryotes, with specific viral proteins containing nuclear localization signals that could prime DNA replication and facilitate genetic information transfer across kingdoms throughout evolution. This highlights a universal role for viruses in shaping the genetic landscape. The intimate associations formed through parasitism and symbiosis also create fertile ground for HGT. When organisms live in close proximity, especially one inside another, the opportunities for genetic material to move between them multiply. Close proximity due to parasitism and cross-contamination due to crowding have been proposed to favor such transfers in both plants and animals. This direct contact or shared environment can lower barriers to genetic exchange, enabling unexpected evolutionary shortcuts. Recent research has unveiled even more subtle and pervasive mechanisms of HGT within eukaryotes, particularly at the cellular level. Beyond the direct transfer of genes, scientists are discovering that cellular components themselves can act as vehicles for genetic information. For instance, apoptotic bodies, small vesicles released from dying cells during programmed cell death, have been shown to transfer DNA, RNA, and proteins to neighboring cells. These bodies can be engulfed and internalized by recipient cells, facilitating the exchange of genetic information. This process has implications for disease progression, as apoptotic bodies can transfer oncogenic DNA to recipient cells and even contribute to the spread of antibiotic resistance genes or viral DNA. Similarly, circulating cell-free DNA, or cfDNA, derived from apoptotic, necrotic, or actively secreting cells can cause genetic alterations in healthy cells, potentially leading to their transformation into cancer cells. Studies on colon cancer, for example, have shown that cfDNA from cancer cells can be transferred to healthy cells, inducing genetic changes. Finally, exosomes, tiny extracellular vesicles, are another mediator of HGT in eukaryotes. They can transfer genomic DNA and RNA into recipient cells, integrating them into the genome and impacting gene expression. These naturally occurring, non-toxic vectors represent a powerful, if still largely unexplored, highway for genetic information exchange within complex eukaryotic systems. Now, let's dive into some truly remarkable examples of how these unseen genetic highways are blurring the lines between vastly different life forms, showcasing the incredible adaptability and interconnectedness of life on Earth.
Consider the transfer of genes from bacteria to fungi. While perhaps less dramatic than transfers to animals or plants, it's a fundamental inter-kingdom leap. For instance, genes have been horizontally transferred from bacteria to common yeast species like Saccharomyces cerevisiae. In another example, plant expansin genes, which play a role in plant cell wall loosening, have been transferred to fungi, further enabling these fungi to infect plants. This acquisition provides fungi with a new tool to overcome plant defenses, illustrating a direct functional benefit from HGT. The exchange between bacteria and plants is well documented, often with significant practical implications. The pathogenic bacterium Agrobacterium is a prime example. It naturally transfers genes to plants, causing crown galls and proliferating roots. This natural process has been famously co-opted by genetic engineers to introduce desired traits into crops, making Agrobacterium a cornerstone of plant biotechnology. Beyond pathogens, ancient HGT events have shaped fundamental plant biology. Land plants and their close relatives, the Charophysian green algae, share a set of glycosyl hydrolase enzymes that were likely transferred from bacteria and fungi to their last common ancestor before plants colonized land. This demonstrates how HGT provided crucial tools for a major evolutionary transition. The transfers from bacteria to animals reveal an astonishing array of adaptations. Take the coffeeberry borer beetle, Hypothemis humcae. Its genome contains a gene called HHMAN1, which strongly resembles bacterial genes and is thought to have been acquired from bacteria living in the beetle's gut. This gene likely helps the beetle degrade coffee berries, a unique dietary adaptation. Even more surprisingly, the Oscar gene, essential for germline specification in holometabolous insects, those with complete metamorphosis, is thought to have originated from a bacterial HGT event followed by a fusion with another genetic domain. Insects, in particular, are rich with examples of bacterial HGT. The adzuki bean beetle, for instance, has acquired genetic material from its endosymbiont Wolbachia, a common bacterial donor in insects. Drosophila ananasi has even acquired nearly the entire genome of Wolbachia pipientis via HGT. Similarly, the psyllid Pachypsilla venusta has acquired genes from its current endosymbiont Carcinella and from many of its historical endosymbionts. These transfers are not trivial. Quantitative insights reveal that out of 1,410 horizontally transferred genes identified in 192 insect genomes, 1,115, 79.0%, originated from bacteria, with Wolbachia, Serratia, Bacillus, Pseudomonas, and Paeonobacillus being prevalent donors. These acquired genes often confer direct functional benefits. For example, bacteria use pore-forming toxins to lyse cellular membranes, and the genes for these toxins have been independently transferred to eukaryotic genomes, possibly playing a role in defense and predation for multicellular eukaryotes. Genes encoding antibacterial imidases and lysozymes have also transferred across the tree of life, augmenting eukaryotic innate immune function. HGT can even influence complex behaviors. A prevalent gene, LOC 105,383,139, transferred into the last common ancestor of moths and butterflies, was shown using CRISPR-Cas9 to be crucial for male courtship in diamondback moths. Furthermore, recent studies in Riptortus pedestris have demonstrated that its Burkholderia symbiont degrades the insecticide phenotrophion into a non-toxic compound using a horizontally transferred gene encoding a detoxifying enzyme, conferring adaptation to environmental toxins. Belloid rotifers, tiny freshwater invertebrates, hold a remarkable record. Approximately 8% of their genes are of bacterial origin, allowing them to survive extreme conditions without sexual reproduction for millions of years. This massive acquisition of foreign genes highlights HGT as a powerful engine for extreme adaptation.
Even in humans, there's a fascinating, albeit debated, possibility of such transfers. The parasite that causes Chagas disease, transmitted by assassin bugs, can insert its DNA directly into the human genome. While the broader extent of bacterial gene transfer into the human nuclear genome is still under investigation, quantitative evidence suggests a more substantial impact than previously thought. For instance, 1,467 human genome regions, totaling 2.6 million bases, across all chromosomes have been found to be more conserved with non-mammals than with most mammals, involving 642 known genes, often enriched with ion-binding functions. The discovery of space invaders, or spin elements, highly conserved between 7 distantly related species, further suggests ancient HGT events. Moreover, lines, L1, the only active transposable elements in the human genome, are known to be involved in the development and progression of various human diseases by activating otherwise silent promoters or promoting deregulated expression of oncogenes and tumor suppressor genes, highlighting a potential link between mobile elements and human health beyond just infectious diseases. These findings underscore our deep interconnectedness with the microbial world and the subtle yet profound ways HGT can influence our biology.
The genetic exchange between plants themselves also defies traditional reproductive boundaries. The parasitic eudicot Striga hermanthica has received a gene from its host, sorghum, integrating it into its nuclear genome. Perhaps one of the most impactful plant-to-plant transfers involves ferns. Around 180 million years ago, ferns acquired a crucial gene called neochrome from hornworts, a group of non-vascular plants. This genetic acquisition allowed ferns to survive and thrive in dark forest environments by enabling efficient photosynthesis under low-light conditions, fundamentally altering their ecological niche. Furthermore, direct observation has shown the transfer of messenger RNA between host plants and parasitic heterotrophs in the Orobanchaceae family, suggesting an RNA-based transfer mechanism that could lead to the integration of foreign DNA. However, this RNA-based transfer hypothesis was not universally validated in a recent study that showed HGTs found in the genome of the parasitic species Cuscuta originated from the movement of genomic DNA, suggesting diverse mechanisms are at play. The movement of mitochondrial DNA between parasitic plants and their hosts is also frequent, blurring the genetic boundaries in these intimate associations. Even between non-parasitic plants, horizontal transposon transfer has been observed, with jumping genes moving between species like rice and millet.
The realm of plant-to-animal transfers provides further evidence of these genetic highways. The whitefly, Bemisia tabaci, acquired a plant detoxification gene, which crucially helps it neutralize plant toxins. This acquisition significantly contributes to the whitefly's pervasive adaptability and its ability to feed on a wide range of host plants, demonstrating a direct evolutionary advantage gained from an unexpected source. From fungi to insects, we see remarkable adaptations. Pea aphids, for instance, are unique among animals for their ability to synthesize carotenoids, pigments typically produced by plants, fungi, or microbes. This ability comes from multiple genes horizontally transferred from fungi, providing the aphids with a unique metabolic capability, which contributes to their body coloration and potentially other physiological benefits. Within the fungal kingdom, HGT is also a powerful force. The production of the potent toxin alpha-amanitin, found in several seemingly unrelated fungal genera like Amanita, Lepiota, and Galerina, is attributed to the horizontal transfer of key biosynthetic genes. This suggests a shared toxic arsenal acquired through genetic exchange. Even large genomic elements can move between fungi, as evidenced by the wallaby element, approximately 500 kilobases in length and containing around 250 genes, which was recently transferred between two penicillium species used in cheesemaking, P. camemberti and P. roqueforti. This element likely plays a role in microbial competition, showcasing how HGT can rapidly spread complex adaptive traits. The boundaries between fungi and oomycetes are also permeable. 4 genes from Magnaporthogresia, the rice blast fungus, were suspected to be horizontally transferred from the genus Phytophthora and are hypothesized to have played a role in the fungus's evolution into a plant pathogen. Conversely, oomycete species like Phytophthora ramorum and Phytophthora infestans have acquired genes from fungi, with these transferred genes thought to be involved in colonizing plant tissues, evading plant immune responses, and breaking down plant cell walls. This bidirectional exchange highlights a dynamic genetic arms race between plant pathogens and their hosts, mediated by HGT. Finally, even among animals, horizontal gene transfer, though considered less frequent, has been documented. A striking example is the smeltfish, which acquired an antifreeze protein gene from herring through a direct horizontal transfer. This genetic leap provided the smelt with a crucial adaptation, allowing it to survive in frigid waters. This rich tapestry of examples paints a picture of life's evolution that is far more interconnected and dynamic than the traditional tree of life model alone suggests. Genes are not confined to their ancestral lineages, but are constantly on the move, hitchhiking across kingdoms, domains, and species boundaries, driven by intricate mechanisms and often facilitating rapid adaptation to new environments or the acquisition of novel traits. This ongoing discovery of unseen genetic highways forces us to continually reevaluate our understanding of how species evolve, how they interact, and what new genetic innovations might be just a leap away. What other profound inter-kingdom genetic exchanges await discovery, further blurring the lines of what we thought possible in the grand narrative of life?
Chapter 4. The Deep Influence of Horizontal Gene Transfer. This final chapter synthesizes the discoveries from previous chapters, allowing us to truly grasp HGT's profound influence on the evolution and adaptation of all life on Earth, and its role in shaping the intricate web of existence. Life's evolutionary trajectory is increasingly described as a complex web or thicket, where genes are not merely inherited vertically, but are also constantly exchanged across diverse lineages. This recognition of HGT as a ubiquitous evolutionary process continually shapes the genetic makeup of organisms. This constant genetic mingling profoundly complicates the notion of a single, universal tree of life. If different genes within a single organism have distinct evolutionary histories, some inherited vertically, others acquired horizontally from distant relatives, then accurately tracing the lineage of a species becomes a complex endeavor. While HGT can blur species definitions and impede robust phylogenetic reconstructions, ancient HGT events have also been recognized as useful clues to organismal relationships, not just confounders.
Beyond simply complicating our phylogenetic maps, HGT acts as an unparalleled engine for biological innovation and the acquisition of novel traits. It allows organisms to borrow pre-existing solutions from other species that have already evolved those capabilities. This borrowing can confer entirely new metabolic capabilities or introduce novel defense mechanisms, driving adaptive radiation and shaping the pace of evolution. While many HGT events are initially neutral or even slightly detrimental, they can later adapt to become highly beneficial to the recipient, demonstrating the dynamic interplay between gene acquisition and subsequent evolutionary refinement. The constant influx of foreign DNA through HGT raises a fundamental question. How do genomes maintain their integrity and size amidst such pervasive genetic exchange? Research reveals that gene acquisition is often not a simple addition of new material. Instead, a common phenomenon during prokaryotic HGT is gene replacement, where acquired genetic material from a closely related strain often replaces existing segments of the recipient genome. This preferential recombination at non-homologous regions, flanked by areas of homology, suggests a sophisticated mechanism for incorporating new genes while maintaining overall genome size and organization. However, HGT can also be detrimental if the gene has no function, is incompatible with the recipient's cellular machinery, or is a selfishly replicating mobile element. Increasing genome size can increase replication time, leading to selection against large, non-functional sections, and expressing non-useful genes imposes metabolic costs. Random insertion can also disrupt existing genes, highlighting the potential costs of uncontrolled genetic influx.
Furthermore, horizontally transferred genes are not randomly scattered across the genome. They tend to concentrate in specific chromosomal regions, sometimes referred to as hotspots. These hotspots, which can constitute as little as 1% of the chromosomal regions in some bacterial species, become hubs of rapid gene turnover. Their distribution depends on local contexts, such as neighboring core genes and the presence of mobile genetic elements. These hotspots act as treasure troves of strain-specific adaptive genes, concentrating most changes in gene repertoires and reducing the potential trade-off between diversifying the genome and maintaining its stable organization. Many mobile genetic elements and antibiotic resistance genes are found in these hotspots, but interestingly, many hotspots also lack recognizable mobile genetic elements, instead exhibiting frequent homologous recombination at their flanking core genes, particularly in naturally transformable bacteria. This localized integration strategy allows for targeted genetic diversification without disrupting the entire genome. Many mobile genetic elements and antibiotic resistance genes are found in these hotspots, but interestingly, many hotspots also lack recognizable mobile genetic elements, instead exhibiting frequent homologous recombination at their flanking core genes, particularly in naturally transformable bacteria. This localized integration strategy allows for targeted genetic diversification without disrupting the entire genome.
This dynamic interplay between gene acquisition, replacement, and localized integration demonstrates that bacterial genomes are not static blueprints but highly fluid and adaptable entities. The integration of foreign DNA often requires sophisticated cellular machinery, including homologous recombination systems which work synergistically with HGT. Cells also possess defense mechanisms, such as restriction modification systems, which provide immunity against foreign DNA and stabilize mobile genetic elements. The selfish operon concept suggests that bacterial operons, which are co-regulated gene clusters, might be a result of selection pressure for functionally interacting genes to remain together during HGT, ensuring their collective benefit. These systems can mitigate the potential fitness costs of newly acquired genes, removing deleterious mutations or non-beneficial regions and optimizing the integration of advantageous genes into existing regulatory networks. This ensures that the acquired genetic traits are not just present but also functional and beneficial to the new host, maintaining genetic efficiency and contributing to the overall fitness of microbial populations. This complex dance between acquisition and integration is essential for the long-term success of HGT, and cells can even evolve to increase or decrease their rate of acceptance of horizontally acquired genes based on the average benefit or detriment. Modern prokaryotes with lower gene loss rates might find HGT, on average, unfavorable and thus evolve to reduce it. Modern prokaryotes with lower gene loss rates might find HGT, on average, unfavorable and thus evolve to reduce it.
The success of a horizontal gene transfer event, whether a newly acquired gene is retained and becomes beneficial to the recipient, is profoundly influenced by the environment. As explored in Chapter 2, it's not just about the transfer happening, it's about whether that transfer actually matters for the organism's survival and reproduction. Genes that are beneficial in one environment might be neutral or even detrimental in another. This concept of gene-by-environment interactions is crucial for understanding the real-world impact of HGT. Studies have demonstrated that the fitness effects of individual genes transferred between bacterial species are highly dependent on the specific physiological environment. For instance, while antibiotic resistance genes are highly beneficial in the presence of antibiotics, they can impose a metabolic burden on bacteria in their absence, making them less competitive. This highlights the unpredictable nature of how environmental conditions impact the fitness effects of each individual gene.
Ecological factors such as population density and spatial structure also affect the success of HGT. High cell density, for example, can induce competence in bacteria, making them more receptive to transformation. In tightly packed microbial communities like biofilms or the gut microbiome, the close proximity of organisms can significantly increase conjugation rates. This means that the local environment can create hotspots not just for gene integration, but for the very occurrence and fixation of beneficial HGT events. Understanding these environmental modulators is key to predicting the trajectory of microbial evolution and, as we saw in Chapter 2, to combating the spread of traits like antibiotic resistance. The human microbiome itself is a prime example of an environment where these dynamics are constantly at play. It's a complex ecosystem where bacteria, archaea, and viruses interact, and HGT is pervasive. The host's physiology, including diet, immune status, and the presence of selective pressures, creates a selective landscape that profoundly influences which genes are transferred, retained, and expressed, ultimately shaping the composition and functionality of our own microbial communities.
While HGT is undeniably more frequent and conspicuous in prokaryotes, its prevalence and evolutionary importance in multicellular eukaryotes are becoming increasingly clear, albeit often operating through more complex and subtle pathways. These transfers often provide significant functional or ecological advantages. For instance, extensive exchange of mitochondrial DNA between plants is well documented, providing the highest known levels of eukaryote-eukaryote transfer. While mechanisms like phagocytosis and endosymbiosis might increase the chance of acquiring new genes in eukaryotes, this is often offset by a reduced need for these genes, because selection often operates on variation not readily generated by HGT in complex organisms. A gene's transferability is also affected by factors like its sequence divergence from the recipient and how many interaction partners the resulting protein has, supporting the complexity hypothesis, which suggests that highly connected genes are less likely to be successfully transferred. Moreover, transferred genes might provide a transient advantage, but are frequently not retained over prolonged periods, suggesting HGT's long-term impact might not always be profound. Nevertheless, the sheer volume of bacterial genes identified in some eukaryotic nuclear genomes, often referred to as inherited chimerism, points to a deep and ongoing history of genetic exchange that fundamentally shapes eukaryotic evolution. On a grander, more philosophical scale, HGT is even postulated to promote the maintenance of a universal life biochemistry, and, consequently, the universality of the genetic code itself. If genetic material can readily move between diverse organisms, sharing successful biochemical pathways and the fundamental language of life, it creates a powerful homogenizing force.
This constant cross-pollination of genetic information could act as a ratchet, ensuring that highly efficient and universal biological mechanisms are retained and disseminated across the tree of life, rather than allowing for radical divergence in fundamental cellular processes. This deep interconnectedness suggests that the very bedrock of life's chemistry may owe its universality, in part, to the ceaseless horizontal flow of genes. The journey through horizontal gene transfer reveals a biological landscape far more dynamic and interconnected than previously imagined. While significant progress has been made, there remains a surprising disconnect between lab experiments and natural environments regarding HGT, highlighting the need for further research into its ecological context. Additionally, further study of neglected eukaryotic groups, such as protists, will help clarify the true frequency and impact of HGT across the breadth of eukaryotic life. This deeper understanding is not merely academic. HGT mediators are already being explored as genetic engineering tools for the selective introduction of desired changes in the genome for gene and cell therapy, opening new avenues for medical innovation. The story of life, continually rewritten by the profound influence of HGT, is still unfolding, promising further revelations about the intricate web of existence.
We've journeyed through the dynamic world of horizontal gene transfer, revealing a biological landscape far more interconnected than previously imagined. From the rapid spread of antibiotic resistance in bacteria to ancient inter-kingdom exchanges shaping eukaryotes, HGT is a powerful engine of evolution, constantly rewriting life's genetic narrative. It challenges the traditional tree of life model, highlighting a fluid web where genes leap across species, driving adaptation and innovation. This pervasive genetic exchange underscores our deep connection to the microbial world and offers new avenues for medical and biotechnological advancements. As we continue to unravel its mysteries, HGT compels us to fundamentally rethink how life evolves and adapts. Thank you for listening to this exploration of life's unseen genetic highways.